Okay, good morning everyone. My name is Juan Jose Velasquez Martinez. I'm a medical student, and today I will talk about an article called Evodiamine promotes autophagy and alleviates oxidative stress in dry eye disease through the P53 mTOR pathway. Perfect. Let's gonna start talking about a really basic concepts like autophagy. Autophagy is a cell degradation process that helps cells recycle damaged components, protecting against stress and restoring balance. This study was focused on Evodiamine. Evodiamine is a natural alkaloid extract from a traditional Chinese medicine plant, known for these anti-inflammatory and antioxidant effects. Okay, dry eye disease is a multifactorial condition that affects between 5% and 50% of the population. Patients suffer from dryness, burning sensation, and in several cases, impaired vision. This disease has a strong impact on quality of life, yet current treatments, such as artificial tears, provide only temporal effects. And finally, oxidative stress happen when too many reactive oxygen species or ROS accumulate damaged cells and promoting inflammation. The general objective is this study aims to explore the therapeutic efficacy of Evodiamine in the treatment of dry eye disease. Right now, we're gonna talk about the different methods used in this article. The first one is called RNA extraction and real-time quantitative PCR. What was it used for? This method was used to measure the expression levels of specific genes in human corneal epithelial cells. What were they looking for? Okay, they want to evaluate how Evodiamine affects the expression of genes related to inflammation and oxidative stress as well as autophagy. And in really basic words, they took out RNA, turned it into a DNA, measure how much of certain genes were active, and compare it to a control gene to make the result reliable. The second one is called Western blot. What was it used for? It was used to analyze the expression of specific proteins in the samples. What were they looking for? With this, they want to confirm change in proteins associated with autophagy, such as LC3, P62, oxidative stress, such as SOD, and the P53 mTOR pathway. In simple words, they break cells, take proteins, separate them, put on a membrane, detect specific proteins with antibodies, see bands, and measure them. The last one is called immunofluorescent staining. What was it used for? It was used to visualize the location of specific proteins within cells, in this case the P53 protein. What were they looking for? They want to observe whether Evodiamine influenced the nuclear translocation of P53, a key step in the activation of autophagy. It involves treating cells, fixing them so they can stay in place, make holes in them so stuff can enter, block unwanted signals, add an antibody to find P53 protein, then add a glowing tag to see it under a microscope. Finally, they stained the nuclei and took pictures. Perfect. Here we have the results. In the first one, they used periodic acid shift staining. The first is the control without treatment, and we see that the more we increase the dose, we see more cycle cells. Here we look to see the polysaccharides and mucins. In the F1, the Western blot technique is used. This is basically for the expression of proteins. In this is a corneal tissue, and it was shown that when EVO is present, there will be a decrease in messenger RNA and proteins, some of which are involved in inflammation and suggest that EVO has a therapeutic effect. In this one, EVO can reduce the rate of late apoptosis after hypolosmolar treatment. And the last one, Evo has the ability to reduce inflammatory cytokines. This one is an immunofluorescence evaluating the nuclear translocation of p53, and we see that Evo increase the nuclear presence of p53, and the PA has the opposite effects. We can see in this one. Perfect. We have the discussion. We had 3 different affirmations of 3 different authors, and we're gonna check if these affirmations are related with our article. The first one, 10, say that Evodiamine attenuates oxidative stress and ferroptosis by inhibiting the MAPK signal to improve bothersome induced peripheral neurotoxicity, and this is related with our article. The second one, Tazdemir said that regulation of autophagy by cytoplasmic p53, and this is as well connect to our article. And the last one, CUM, said that the Evodiamine prevents traumatic brain injury through inhibiting oxidative stress and via PGK1 and RF2 pathway, and this is as well connected to our article. Finally, we have the conclusions. The first one says that it provides objective tools for measuring the effectiveness of a treatment. Molecular biology offers quantitative and reproducible methods for evaluating how and why a drug works. And the second one says that it facilitates the identification and validation of therapeutic targets. Molecular biology allows for the identification of precise targets for the development of new drugs, surpassing treatment that only relieve symptoms such as artificial tears. Thank you.